So today is the 12th. We've got 1 week until our first exam, so that's a week from today. I will be during our regular class time. I'll talk a little bit more about it on Monday. Um, on Monday, the 17th, um, I will kind of give you a rundown of what to expect on the exam. You guys have the practice exam, and I think that's a good place to begin kind of reviewing, seeing the questions, like the types of questions. I'm going to, I've kind of used that as my starting point for writing the exam. And I'm going to, go back and look at some questions and be like, oh, this is a good one. And then other ones I'll adjust. And then if there's additional content, for instance, questions from today's lecture, um, I'll let you guys know kind of like a final whenever I have like a final edition of the exam, I will let you guys know like here's the distribution of the content. But we will talk more about the exam on Monday. Um, I've got I've got some availability for office hours tomorrow or Friday, so please just contact me. I don't have set office hours. I'll have my set office hours on Monday at 12:30, but if you want to meet before then, by all means. Um, I dropped the ball this week on getting assignments and activities for you guys, and I just kind of didn't completely drop the ball, but kind of did with respect to your quizzes. I added two more quizzes today, but I went ahead and set those due on Tuesday the 18th. Um, One thing about those quizzes, if you want to meet with me during office hours and just kind of walk through them and work on them with my assistance, that's fine. Because those quizzes are not quizzes in the sense that tisk tisk kissed, you better, oh boy, this is a test. No, they're basically homework. So if you want to meet and talk about those, I'm happy to do that. Um, Assignments, in order because I kind of previously have promised you guys, yeah, you're going to have this many grades. I'm just going to have two activities for this week that are going to be freebies, so I'll be just adding those. They'll be numbered just in sequence, but there's nothing for you guys to actually submit. Um, then the last other thing was participation on Monday. I did not put the discussion option up. So what I'm going to do for Monday to reflect on Monday is if you were here and you participated in the polls, you're going to get those 5 points. If you were not here and you are going to do the discussion, then I'm going to leave a blank in the grade book for you. So what that means is that it's not going to count against you. It's just going to reduce your overall total possible points. So it's not going to, I mean, it's not going to sway your grade really one way or the other dramatically. So I just wanted to address all those things. Now, before we start talking about protein purification, which is, I think it's a fun topic because I think that it's helpful to kind of understand like biochemistry as a field. Um, but before we do that, what I wanted to do is I wanted to throw some poll questions your way, specifically regarding inhibition. And so I want you guys to be able to think about inhibition in a couple of different ways. There's basically four graphs, but then I want you to not only be able to visualize those graphs, I want you to think about, well, what are the ramifications of this or this? So one way that I'm asking that is with this first poll question that I'm posting right now. So the question reads, which type of inhibitor's data intersects with uninhibited data on the y-axis? So the inhibitor's data, so that means the enzyme's activity with an inhibitor present. So what type of inhibitor is going to do that? Um, for me personally, what is most helpful whenever I look at a question like this is to imagine, like, to kind of mentally draw that out. So what I'm looking at there is, I'm looking for a y-axis, and what I'm going to do is I'm going to go ahead and draw my double reciprocal plot. Nope, that's not it. Um, my double reciprocal plot there and there. So 1 over initial velocity, and then 1 over substrate concentration. And I could draw that in the middle. It's force of habit that I always draw it on the far right-hand side. Um, sorry, just a second. I see something in the chat. If you're on the mobile app, then you might have to tap your screen. I'm not exactly sure why that happens, but we're going to have another poll question in just a second. If you can find the menu, or the controls for your Zoom meeting, that might also be helpful. But please don't cry so much that you float away, which is what appears to be happening with your emoticon there. It's going to be okay. Okay, so we've got that right there. So this is no inhibitor. And then I'm going to go ahead and switch colors. I'll go with green. Plus inhibitor. We're having an intersection on the y-axis. And so there's there and there. So this is plus inhibitor. Now I'm going to go ahead and end the poll and I'll share the results. 66% of the class responded correctly with a competitive inhibitor. So a competitive inhibitor has that intersection of our lines on our y-axis. The others are going to intersect in different places. Now to kind of rehash those real quick, just because I think that it's helpful to do that. I'm going to go with another color. So let's see, I'm going to go ahead and write. I'm going to put in parentheses comp. Then I'm going to switch colors and we'll go with, let's go with blue. Well, where is a blue one going to intersect? This is plus inhibitor. If we see the intersection, that would be in quadrant 2 is one of the ways that I mentioned that. That would be mixed. And then I'm not going to make this too noisy of a slide because I'm not going to draw one of them. Our last one is right here, which is a good way to lead into our next question. And this is a non-comp. The one that I've elected to not draw is an uncompetitive inhibitor because the uncompetitive inhibitor, your plus inhibitor data does not intersect at all. So those two lines run parallel to one another. Okay, I'm going to stop my share. I'm going to give you your next poll question. Back, clicked back, and now I'm going to Q2. Question 2, not Quake 2, the computer game. What type of inhibitor drives Vmax down but doesn't change the Km? So when you look at this graph, and I'm sorry for talking over your thoughts, but I'm just trying to kind of coach along here. Think about what your intersections, or what is the significance of the different intersections? Your y-intercept is what's known as your 1 over Vmax point. Your x-intercept is negative 1 over Km. And so we're definitely trending in the right direction. That's where I'm going to stop that. And I noticed as I said that things were going in the right direction, there was one answer that people started, that got a little bit popular. Non-competitive and uncompetitive are not the same thing. I did not draw the uncompetitive. Okay, so I'm going to go ahead and end the poll here. We've got, we got 63 out of 63. Ooh, sounds like we got 100% participation. That's a bonus point. And you might think to yourself, wow, that sounds kind of arbitrary. Yeah, absolutely. But I try to do things that are arbitrary and subjective in your favor, as opposed to like arbitrarily doing something mean. So the correct answer for this was a non-competitive inhibitor. And I think that it's good to be able to kind of come up quickly with this graph right here, these 4 different lines, and then just kind of recall, okay, in uncompetitive, we have no intersection. So here we've got the non-competitive that intersects at the x-axis. I always kind of think about this as y intersection, quadrant 2, x intersection. Competitive, mixed, non-competitive might be helpful, but also just kind of familiarity. And the more that you see it, you're going to get more comfortable with it. All right, now I'm going to go back, and now you can't see my poll questions any longer, which is perfect. And what I'm going to do is I'm going to toggle ahead to talk about protein purification. Protein purification is finding a needle in a haystack. You have a very complex mixture and you want to pull one protein out of there. Biochemistry is a field and sciences are very reductive, and not reductive in the oxidation reduction fashion, but instead like, let's take something complex and let's isolate one thing from there. Now, the kind of topics that we're going to be investigating, and if you're thinking, man, Joel got really good with the PowerPoint this week. Yeah, I did get really good using Canva again. We're going to be looking at separation, so separating components, then analyzing, and quantitation. So think about separation as, think about our target as we want to purify a protein. Now, I think that it's pretty common for people to think of like, okay, well, what's a protein? I mean, it's very common. Like if I went up to, I don't know, my neighbor and I was like, hey, think of a protein, they'd be like, steak. And I'd say, well, not really. That's not where I'm going. Um, so I think that it might be helpful to identify a protein. And so I think it might be helpful for everyone, if there's one at the top of your head or something like that, go with that. But if not, maybe like hemoglobin or hexokinase, a protein or an enzyme that has a particular function, a specific function. And, if you think about hexokinase, for example, that's a protein that's found in the cytosol in a cell. I always kind of think about a cytosol as being like, it's the ocean of the cell. Is there a bunch of proteins in there? Yes, there are. So essentially what we're going to be doing is we're going to be talking about taking that, I'm going to use hexokinase. I'm going to try to come back to that over and over again. We're going to take not just one cell, but tissue, like, muscle tissue or liver cells, and we're going to grind them up and we're going to try to pull hexokinase out of all of those cells. So that's what we're aiming to do when we talk about separation. Then the analysis is a question of, well, how effective were our separations? Did we get down to just hexokinase, or is our protein in a very, did we go from a very messy mixture to a, well, slightly less messy mixture? And then quantitation is a way of saying, well, how much do you actually have? Now, if I can give you another kind of analogy, when it comes to protein purification, what I always like to do is I always like to think about, let's say that you had a beaker, and I'm sorry, but I just couldn't resist staying close to a science thing. So we've got a beaker, and let's say that beaker is filled with M&Ms. And then let's say that it's filled with peanut butter M&Ms. And then let's say it's filled with Skittles and peanut M&Ms. And you kind of get the point there. So we've got all of these candies. What make those candies, or would everyone agree that they are all candy? Not a hot take, not a controversial idea there. Okay, good. We got lots of yeses. Can anyone tell me about what makes them different from one another? Is there anything that makes these different from one another? Type, flavor, presence of nuts. Some have chocolate, yeah. Their shapes are going to be somewhat similar, but also somewhat different. Like, I feel like I can pick up a Skittle and be like, that's a Skittle. But if you hand me an M&M, I'm going to be like, yeah, that's an M&M. That's not a Skittle. Skittles are a little bit, in my recollection, they're a little bit chunkier. Colors, absolutely. Skittles are going to have a little bit more robust or like a brighter color to them. So although there are some things that make them different, there are some things that make them very similar to one another. Now, if you can imagine that a mixture of texture, absolutely. We've got a mixture of all of these different candies. And let's say that, oh, letter, absolutely. Let's say that you're like, I only want to eat green M&Ms. Then that would be like you're on tour and that's your what is it called? Your rider. Whenever you get to your next city and you're like, we only eat green peanut M&Ms before we go on stage. You know, anyway. So you've got if you only wanted those green peanut M&Ms, then actually separating out and isolating those green M&Ms, there's lots of steps. Like if you were able to enrich for just green candies, well, you could definitely tell that a green Skittle is different from a red M&M. But if you collected all of the green candies, you're going to have some similarities and some differences there. That's kind of how protein purification works also, where we've got some very similar properties, but then there's like shades of difference to them. So I think that it's helpful to kind of visualize that and just imagine that, well, when you're going through the process of isolating for just your candy or just your protein, one of your aims is going to be to um, enrich for your specific protein. And what I mean by that is, let's say you start out with 100,000 different proteins, and let's say that 2,000 of them are your protein. Okay, so you're looking at about 2% of the total protein is your protein or your candy, whatever. And then you go through a bunch of different separation techniques and enrichment steps, and you end up with, well, 100 proteins. Let's see, 100. No, I gotta change that. Sorry. 175 top prot total proteins. 175 total proteins. But 100 of them are yours. Okay. Um, have we enriched for our protein of interest? Yeah. Did we lose some of our protein along the way? We did. But if you think about the ratio of what our protein is relative to the total amount, oh, we've done a great job. More than 50% of the total protein is our protein. So keep that in mind anytime you think about or talk about protein purification. Now, a handful of y'all are going to take Chem 3276. That's kind of the lab companion class to, well, this lecture. And so you'll talk about protein purification and you'll try to purify a protein. You'll do a couple of different things. So what we're aiming to learn about today are different types of chromatography. Put a yeah or a emoji into the chat if you've heard of chromatography or have you done chromatography before. Fantastic. Okay. We're going to talk about SDS page. Some of y'all have maybe talked about or done SDS page. We're going to do a Bradford assay. We're going to talk about UV detection of proteins. And kind of the first one is we're going to talk about salting out. Now, how can you analyze a protein if it's part of a very complex mixture? It's it's involved, it's an involved process, but the first thing that you really want to aim to do is get rid of all of the other junk, all of the fluff. And so that's where we get to separation. So separation is how do we get the one thing that we actually care about out of a complex mixture? And I did a Google image search for needle in a haystack and I found this one. And so unfortunately, that kind of misses the point of that phrase, but it's fun. We're trying to find that needle. Our hexokinase is our needle. Now, the very first thing that you do whenever you are trying to isolate a protein is you've got to have a source for that protein. And so in this particular case, we're going to go with we got a mouse liver cell or mouse liver. That mouse liver is our tissue, and that tissue is made up of tons of different cells. Now, the thing that we want to do and that we have to account for, the couple of things that we want to account for are we've got to take this liver and has anyone add to the chat or signal that you have used a mechanical pipettement before? Okay. Mechanical pipettement, a handheld pipettement. Um, there's three different volumes. One of them is a milliliter, one of them is a cup like 200 microliters, and then one of them is about 20 microliters. Um, there's a little bit of a skill and a little bit of a learning curve to using one effectively. Um, can I see anyone who has ever tried to pipette a solid and had success pipetting a solid? Add to the chat of yes, or I'm not seeing any yeses. I'm seeing a straight up no. One of your classmates gave me a furrowed brow. Absolutely. Pipetting a solid. Yes. What? Pipetting a solid is virtually impossible. So if you like, let's say that somebody brought you, hey, here's the mouse liver, and we're going to try to purify this protein from here. And you got the mouse liver, and you're like, ugh, it's not working. You're right. It's not going to work. So what do you have to do? You have to make that solid. You have to make it a liquid, something that you can actually pipette. I hope that clears up the confusion there, Trinity. Um, you have to homogenize it. You have to take that tissue and make it basically like a kind of like a smoothie almost. Now, is that an appetizing smoothie, especially considering that some of y'all are eating your dinner at this hour? Not really. And I'm sorry for giving you that visual, but there are a couple of different ways that we homogenize tissue. One way is with this right here, the same way that we make tortillas, or, well, compounding for like pharmacists and stuff. We use a mortar and pestle. I was a graduate student and worked on lots of proteins from plants, and so that's what I would do. We'd freeze them with liquid nitrogen and then we just grind the heck out of them. Um, Another way that you can homogenize tissue is if you look here, this is a microcentrifuge tube. Chances are it's about a milliliter and a half, and there's a bunch of small beads there. And then what you do is you just shake it at a very, very high speed, and those beads, they're just going to basically, it's almost like they're little bullets flying through the tissue that you have. And you're going to shake it so much that that tissue kind of makes a homogeneous solution. That's what you need to do. And at that point, you can work with that. You can pipette that up and down. So we've taken a solid, added some sort of buffering system or buffering agent to it, and then, well, blended it up. We've ground it with a mortar and pestle. Sometimes you will blend things with a blender. In 3276, chances are you're going to use a blender. Um, Okay, so the first thing that you need to do is basically make this tissue something that you can pipette. Now, if you're working with like blood or saliva or something like that, you don't have, it's a different treatment. But you have to think about what the source is that you're working with. Um, Now, what you're then going to do is think about separating the different proteins from your mixture. What properties do you think that that protein actually has? In order to kind of think about that, you got to think about, well, what is a protein made up of? Give you a hint. It starts with an A and ends with a amino acids. Absolutely, amino acids. Amino acids, do they have unique properties? Are there some amino acids that are positively charged, negatively charged? Absolutely. So the properties of every single amino acid contribute to the properties of a protein. Now, one thing that I want to bring up right now, which is going to be valuable after a little while, is when we talk about molecular weight or size, we can say that the average amino acid, 1AA is what I always like to use as my shorthand, is approximately 110 Daltons. A Dalton is the same as 1 gram per mole. 1 Dalton is 1 gram per mole. So the average amino acid is about 110 Daltons. Sometimes you'll see that as 107. Do amino acids differ in their molecular weights? Absolutely. But we can operate under the idea that the average amino acid is 110 Daltons. You might also see this listed as 0.11 kDa or kilodaltons. Okay, so if you have something like if you have a polypeptide that's 10 amino acids long, that 10 amino acid long polypeptide is about, well, 1100 Daltons or 1.1 kilodaltons. If you have a peptide that's 100 amino acids, well, then you're talking about close to 10,000 Daltons or 10 kilodaltons, sorry. So the properties of every amino acid of those R groups are relevant for separating different proteins from one another. Now, the most common properties that we use to separate things are solubility, the charge of any ions, the polarity, the size of your protein, and binding specificity. So that binding specificity, think about that as does this protein have the function to bind to a particular molecule? So as an example, the protein or the example protein that I'm going to use is hexokinase. What hexokinase loves to do is it loves to bind onto glucose and metabolize it and phosphorylate it, do something to it. So it has that unique property. Okay. Now, um, for these different properties, for these different separation techniques, or sorry, for these different properties of proteins and amino acids, we have different techniques that we can use to separate based on. The first one that we're going to get after, and this is kind of a preview of what we're going to talk about today, salting out. Then ion exchange chromatography. We're going to also talk about isoelectric focusing and electrophoresis. Some of you have probably talked about if you've taken genetics, and I've actually heard that in recent years in like an AP bio class, you do DNA gel electrophoresis. I don't know if anyone took AP bio. I didn't. Miss Abel discouraged me from doing that. But we'll talk about electrophoresis. It's a way of, well, sending an electric current through something and getting them to separate. You can separate based on polarity, the size of a molecule, and binding specificity. So this is a good preview of all the different separation techniques that we're going to talk about. So the first one that we're going to do, and this is kind of a roadmap for the day, is we're going to talk about salting out. Now, in essence, like I'm going to draw a nice picture right here. And is there, and then there. I'm going to call this a pellet. And then if that's the pellet, what would you call this right here? Anyone add an idea or propose a name for that? And as opposed to a pellet, oh, okay, solute. I'm gonna say, ooh, supernatant. That's what I was looking for. Superman. Okay, yeah. Um, supernatant. Now, if there's one thing that I remember from Gen Chem 2 labs, um, there was always the conversation of, oh crap, I didn't read the procedure. Do I get rid of the supernatant or the pellet? And then someone would be like, listen, I'm like 5 steps ahead of you. You get rid of the supernatant. And then it's like, okay, cool, supernatant, toast. And then you watch them for a couple more minutes and you're like, you're behind me. And then they're like, yeah, that's what I said. I was talking to someone else. And you're like, no, I've done more work and I listened to you. Why do I ever listen to people? So you have your supernatant and your pellet. Do you get rid of the supernatant? Do you get rid of the pellet? Now, at the end of the day, what this boils down to, supernatant versus a pellet, what we're talking about is soluble versus insoluble. If a protein is soluble in a solution, where do you think it's going to be between those two names, the supernatant or the pellet? The supernatant. Perfect. If it's insoluble, it's going to go to the pellet. It's going to precipitate. So think about those terms, soluble versus insoluble, and insoluble, kind of think about if something's insoluble, it's in the pellet, it's also considered to be a precipitate, or it has precipitated out. So we've talked a little bit about protein solubility previously. Does anyone remember the context that we talked about solubility? I'm thinking of close. I'm thinking of two letters. The first one is P. PI. Close. H and I are close to each other on the keyboard. So PI. What is PI? Perfect. PI is also known as our exactly the isoelectric point, the neutral point, where our molecule has an overall charge of zero. That is where our protein is least soluble. Okay, so just to consider this, if you had your protein in a solution and you knew exactly what the PI was, and you adjusted the pH to your protein's PI, and then you centrifuged it for a second, do you know where your protein is? You should have solids. Your pellet. Your protein is now in the pellet. So if your protein is soluble, but then you adjust the pH a little bit, and you adjust the pH of your solution to exactly what your protein's PI is, boom, it falls out of solution. The great thing about that is you know exactly where your protein is. So in that case, would you save the supernatant or would you save the pellet? Save the pellet. Get rid of the supernatant. Does the supernatant have lots of proteins in it? Yes. It's got lots of proteins that aren't your protein. So just to kind of put it in that similar sort of terminology. Okay. So salting out is a technique where you can precipitate proteins. And when you are going through a salting out procedure, you don't necessarily know what concentration or what is going to get your protein to precipitate out. So what you will do when you are doing a salting out technique is you're going to add a salt, typically a salt or the salt that you usually add is NH4 2 SO4. Does anyone, perhaps anyone who's like an LA or an SI for Gen Chem 1, does anyone remember that compound? I saw at least one person say it. Yep, ammonium sulfate. So ammonium sulfate. You add that to your solution, and then you add a little bit more. Add it very gradually, and then you're going to centrifuge. And there are going to be some proteins that precipitate out, and they go to the pellet in the presence of that, and then others that are going to remain in the supernatant. Now, with that in mind, I want you to think about any technique that you do when it comes to protein purification is you're learning something. Is the objective, let's say that I'm not going to go far into quantities, but you add X, I'm going to say X molar ammonium sulfate. I'm going to say AM sulf. That causes some proteins to go into solution. And let's say hypothetically that your protein does not go, or sorry, some proteins to go into the pellet. Let's say your protein does not go into the pellet. Was that a waste? No. The reason why it wasn't a waste is because let's say that we add this amount of ammonium sulfate and 25% of the total proteins. Keep in mind, we've got this tissue that we blended up. We've got hundreds of thousands of different proteins. 25% of the total proteins go into a pellet. And your protein doesn't go into the pellet. Oh, dang. Is that good? Is that bad? Well, if your protein's not in the pellet, do you know where your protein is? It's in the supernatant. Okay. So, one, we still know where our protein is. Two, what is our goal here? We're trying to take a very complex mixture and we're trying to get rid of junk. When you look at that right there, wherever you're looking on your screen, we've gotten rid of 25% of the total proteins. Have we cleaned up our sample? Yeah, we have. This is an instance where you can be all Pollyanna-y about it and be like, hey, everything is wonderful. So what have we done? We've gotten rid of a bunch of junk. Do we still have like, you know, of the 75% of the remaining amount of protein, our protein's still a small fraction, but we cleaned it up a whole lot. It's almost like I could think of any number of analogies, but I'm just not going to. Okay, so we'll just proceed from there. So you enriched. So this is a technique that's beneficial even if your protein doesn't go into the precipitate. If it stays in the supernatant, you have these kind of binary questions. Where is my protein? Is it in the pellet? Nope. Okay, well then it's in the supernatant. What amount of protein have I gotten rid of? Oh, I've gotten rid of 10%. Okay, was this effective? Yeah, for getting rid of 10% of the protein. Great. Have I enriched for my protein? By virtue of getting rid of 10% of the protein, yes, you have enriched for your protein. So try to think about it in that way. Okay. Now, does anyone have any questions about basically homogenization or salting out? These are the two techniques that we've begun, that we've started. And what we're trying to do is get our tissue into solution and then get rid of crap. Okay. You can send a private message or just speak up. Now, next up, chromatography. There's several different flavors of varieties of chromatography. I included this picture here on the right because, well, it's a type of chromatography that, one, it's fun and colorful. Two, it's something that you might be familiar with. There's paper chromatography, column chromatography. Chances are, I know that kind of a classical organic separation is to separate fluorine from fluorinone. I don't know if Dr. Rastogi has you guys do that or not. Okay, we're getting nods of yes. Anyone who has done fluorine versus fluorinone, can you tell me the property that you're separating based on? Okay, I got a no, and then okay, it's a ketone. We've got fluorine and fluorinone polarity. We're separating based on these two different compounds having different polarity. Great job. So that's one property that you can take this mixture and separate these two components. So this is also, you can do this with different sorts of dyes. If you remember in any sort of chromatography, there's your mobile phase, there's your stationary phase. I've always thought that with paper chromatography, it's a little bit clear, like, okay, well, the part that's not moving, that's going to be my stationary phase. So my paper is my stationary phase. What's my mobile phase? Well, that's the liquid that kind of uses capillary action to travel through my paper. Column chromatography that we're going to be talking about is one place where people get a little bit mixed up. and they kind of blur the lines. Well, that's not moving, that is moving. Um, Whenever you're doing column chromatography, because if I remember correctly, the fluorine fluorinone separation is done with a column. Can't remember exactly because one of them's like light yellow and one of them's kind of greenish. I can't remember exactly. Um, but in order to separate those two things on that silica column, okay, fluorinone is yellow. Okay, thank you. Um, you can use, well, you add a solvent. And you use that solvent to drive the either more polar or less polar substance through. You add a solvent, and then you just kind of wait and you collect different fractions. Now, for biochemists, do biochemists do paper chromatography? They do. Is paper chromatography good for analyzing proteins? Not particularly. Paper chromatography is good for analyzing um, sugars, lipids, small volatile organic compounds, organic compounds in general, not just volatile ones. Um, Typically, when a biochemist is doing chromatography, they're going to be doing column chromatography. And with column chromatography, most often, what they're going to be doing is separating based on your charge, molecular weight, hydrophobicity, or affinity. And we're going to talk about each one of these. Um, but I will put little pluses by all the ones that kind of are the most common for a biochemist. Now, with that in mind, do we still have to navigate or deal with the mobile and the stationary phase in column chromatography or for a biochemist? We absolutely do. Now, here is a kind of time-lapse progression of separating different proteins. So if you look at this vessel right in the back versus this one in the foreground, the one in the back is kind of the way that you started out. Then you give it time, and well, sure enough, those proteins begin to separate. Now, when you look at this, and I'll draw your attention primarily to the one in the foreground, just because they all kind of say a little bit of the same thing. But what do you think is kind of factually, or what do you think is inaccurate about the picture in the foreground? And if you say, well, it doesn't have a ring stand, you're right, yeah. Colors, absolutely. Very rarely does a biochemist have a protein that actually produces a color. So whenever it comes to purifying a protein, you don't really have the opportunity, like the fluorine fluorinone separation, you kind of watch that. Biochemists, they kind of watch it and they're like, well, it's still clear. Yep, still clear. Oh, look, I'm done. Cool. My clear solution started clear, ran clear, and 75 fractions later, here we are. We're all done. So there are some ways that you can kind of navigate that and detect proteins, and we will revisit that. But this gives kind of the rundown that you have a protein sample. That's your mixture of different proteins. That is your mobile phase. That is going to go through this solid porous matrix. I'm going to refer to this as your beads. And if anyone's ever watched the TV show Arrested Development, which kind of fell out of favor about 15 years ago, but here we are. I'm still using it as a reference. Anytime someone in that show says beads, someone else says bees, and then there's hijinks, shenanigans, and hilarity. So beads, these are molecular beads. I always kind of think about them basically as like small grains of sand almost. So those beads constitute our stationary phase. Now, this protein sample, I'm also going to refer to this periodically as your buffer or your flow through solution. Okay, and then at the bottom, we've got this porous support so that the beads don't flow through and that basically everything stays in that column. Now, the first type of chromatography is IEX, also known as ion exchange chromatography. Now, ion exchange chromatography, well, an ion is just a charged species. A cation is a positively charged species, and an anion is a negatively charged species. Now, for a cation exchange chromatography column, I want to draw your attention out here. Cation exchange, this is one of the parts that's a little bit confusing. The beads are negative. If the beads are negative, well, what's the charge of the protein that we're looking to bind? Positive. Absolutely right. So in cation exchange chromatography, I always kind of think about this as similar to when you go fishing, what are you trying to get? You're trying to get a fish. When you go cation exchanging, what are you trying to get? A cation. So our beads bind, I'm just going to put a positive symbol there, positively charged proteins. Oh, man. Can anyone think of any amino acids that could contribute to those positive charges? Maybe something like an close. That's asparagine. Asparagine's not histidine, arginine, lysine. Those are three characters that are going to be positively charged. Okay. Philip, you got ahead of me because when we talk about an anion exchange column, well, our beads for an anion exchange, those beads are positive. So those beads will bind negatively charged proteins. So Philip said it. We're looking for proteins that maybe have a lot of aspartates or glutamate residues. Thank you very much for that. Okay, so the terminology, cation exchange, anion exchange, that tells you what the bead is looking to bind to as opposed to what the charge of the bead is. So just keep that straight because I know that I struggled and I fumbled with that a handful of times. Okay. Now, if you have a mixture, as is evident here, we've got a bunch of proteins. Some are positively charged, some are negatively charged. Do you think that the makeup of your positive and negatively charged proteins will differ based on the pH of the buffer that you use? Absolutely. So when we do our homogenization of our tissue, if we do it in a buffer with a pH of 3 or the pH of 9 or a pH of 7, the number of positive or negative charges is going to vary a little bit there. Now, with that in mind, we'll get our mixture of proteins. And let's say that this is let's see, I'm going to look at the actual example that has the words on it. Polymer beads with negatively charged functional groups. Okay, so we're going to add our mixture. We're going to let that flow through. And then we're going to put this bulb up here with our buffer. The same buffer that we used to homogenize our tissue to begin with. Does everyone follow me there? Okay, so we blew up the cells and now we're throwing it through. So our proteins are migrating through. If our beads are negatively charged, what do you think is going to happen to negatively charged proteins? Our beads are negative, proteins that are negatively charged pass right through. You're absolutely right. They're repelled. They're going to move through that column. They're not going to slow down. Now, our positively charged proteins, what do you think they're going to do? Well, they're going to do the opposite. They're going to grab on. And no, your internet connection did not get slowed down. They're just going to grab on because they have that opposite attractive force. And if you think back to column chromatography in organic, if you played the waiting game, if you ran that solution, that solvent through over and over and over again, eventually those molecules would migrate through. Well, in this case, in ion exchange chromatography, if our beads are negatively charged and our protein is positively charged, it's going to grab onto that bead and it's going to stay there. It's going to stay there until you basically give it a reason to leave. So with negatively charged beads, our negatively charged proteins go straight through. Positively charged proteins grab on and stay on until they have a reason to leave. One reason to leave would be if I've moved my stuff around. Okay, there we go. If our buffer pH changes. So if we make our buffer that is flowing through our column more acidic or more basic, well, now all of a sudden, those proteins that are bound to those beads, well, some of those residues might get deprotonated. In which case, they're like, well, my structure has changed ever so slightly. I'm going to move on. So, one way for this point right here, how do you get bound proteins released from the column? Well, would we use acidic because it not necessarily that, but instead, um, this is all about ionization of those amino acid side chains or those R groups. So if you have, let's say, a glutamic acid and aspartic acid, or sorry, if you have a bunch of lysines and arginines that are mediating the interaction between protein and these beads, I'm going to say R and K residues. If you have a bunch of R residues and K residues that are mediating that interaction, then you make the solution more acidic or more basic. Basically, shifting one way or another, the ionization of these amino acids, that R or that K, that's going to change. They might become more, or they might become slightly less protonated. Does that make sense? Okay. So now what we've got is um, this question of how do we get proteins to basically release from our column? One way is change buffer pH. Okay? Because when we change the pH of our buffer, we're changing the ionization state of our amino acids that make up our protein. Another way, well, these beads right here, those negatively charged beads, what are they looking for? They are looking for a positively charged ion. They are looking for a cation. Right now, we've got a protein that provides that cation. What if that rolled through? One of your classmates just gave like, oh my gosh, what a controversy. They gave shocked eyes. Yeah, you're absolutely right. If a sodium ion rolled through, those beads don't care. Those beads are negatively charged. They're looking for a positive charge. And they see that ion, they're like, ooh, yes, perfect. I'm going to bind to that ion. And what is that going to do? That's going to displace the protein, the positively charged protein that was there. Okay. Um, so you can change your buffer pH or add salt. So those are your ways to elute a protein. And both of those work whether you're talking about an anion or a cation exchange chromatography column. All right. So now we're going to go to our next chromatography technique. So we've got ion exchange chromatography where we're separating based on charge. Next chromatography technique. Oh man, look at that. Those images kind of look the same because they are kind of the same. Slightly different color pattern. This is gel filtration chromatography, also known as GF chromatography, not to be mistaken with girlfriend or gluten-free chromatography. Gel filtration chromatography is a technique that separates based on molecular weight, separates on size. Gel filtration chromatography is also called size exclusion chromatography, or sometimes people will call them molecular sieves. I never call it molecular sieves because it has flashbacks to 10th grade English class where I could not pronounce that word, and don't worry, Miss Rhodes made me feel great about that. Here we are, though. Molecular sieves. If you know what a sieve is, again, 10th grade English class, the sand flows through your fingers like a sieve. It's basically, I always kind of think of like a cheese grater. It's got a bunch of small holes in it and you can separate things. Now, the great thing about all of our column chromatography techniques is we're dealing with beads. These beads are insoluble little things. What makes these beads different from our ion exchange chromatography beads is they're uncharged. Okay, so proteins are not attracted to those beads because of an opposite or a complementary charge. Instead, they are porous. They've got little holes in them. Now, the way that, so proteins do not bind to these beads, they just kind of move around or through or with them. Your largest proteins are going to elute first, your smallest proteins will elute last. How do you get bound proteins released from the column? You just wait. So gel filtration is a practice and is an exercise in patience. Now, if I can give you kind of an analogy of what I imagine or what I always visualize whenever it comes to protein purification doing gel filtration. What I've got is a coffee cup here. This coffee cup has a bottom to it, so it's not a perfect example. I've also got this robot that I'm still building from one of my kids' Christmas gifts. A small piece of plastic broke. I'm waiting for that to arrive in the mail. These are my two proteins, a baseball and this robot head. Okay, this coffee cup is my bead. Now, let's say that would everyone agree that these two are different size proteins? And oh, look at that. That's perspective right there. Okay, now they're on the same plane. The baseball is smaller than the robot head. The robot head is my large protein. What a protein kind of, and I'm projecting here, what it wants to do is enter this bead and go through it. Now let's watch the robot head. Does it enter it? No, it cannot enter and go through. So what happens is it basically, think about using basketball terminology, it rims out. Boom. So our large proteins never enter the bead and instead they just kind of go by the wayside. Now compare that to our baseball, our smaller protein. What's going to happen is this baseball doesn't go straight through, in part because it has a bottom, but also what it's going to do is come in contact with the bead and then watch, my hand is blocking all of that. It's basically going to go through, and now my finger is going to act as it, and circle and circumnavigate the interior of that bead. So the smaller the protein, the longer that it takes. So now if we used okay, now we've got two different proteins. One is a pencil sharpener, one's a baseball. Both of these proteins will come in contact with our bead, but watch what the pencil sharpener is going to do. It's going to hit the interior and it's going to be in contact with the interior of the bead the entire time. It's not just going to jump on one side and slide down. Instead, the way that it migrates through is it's almost like it's going through a spiral staircase. So there's a relationship between the size of the protein and the rate that it takes as it moves through that column. So in gel filtration chromatography, basically the most important, or your takeaway from this, is your largest proteins go through fastest. Your smallest proteins go through more slowly. Now, one thing that's important to recognize about this is gel filtration chromatography in practice, you're using proteins that are like 15 kilodaltons and 10 kilodaltons. So that's a difference of approximately 50 amino acids. The more narrow the range in protein sizes, the better resolution and the more effective this is. But when it comes to gel filtration chromatography and separating different components, you just sit and wait. And then I hope that everyone follows this right here. You just collect different fractions. So this 1 is the very first solution that came off of the column, 2 is the second, 3 is the third, 4 is the fourth. Okay, so now, does anyone have any questions about our first 2 types of chromatography? Well, kind of 3, almost. Okay, if you do, go ahead and add them. Otherwise, proceeding to affinity chromatography. And affinity chromatography is your protein has a function. It has a binding ability. So explain again how the biggest molecules go through faster. Absolutely. So the biggest molecules are going to go through faster because whenever I say through, I mean through the column. They're not even going to go through the bead. So it's almost like they just move on past. They hit the rim, gone. They don't even stay around. Whereas this pencil sharpener comes in contact and then it has to migrate through very, very slowly because it's coming in contact with the entire interior surface. Does that make sense? Okay, great. So affinity chromatography. We have our protein of interest and a ligand. A ligand is the thing that our ligand is the thing that our protein is capable of binding to. So our protein of interest, I'm going to say HK, that's that hexokinase, and the ligand would be glucose. Hexokinase and glucose, those are completely examples. Those are just examples. That ligand could be any number of other things that attract any number of other proteins. Now, when it comes to affinity chromatography, are we still worrying about beads? Are we still thinking about beads? As my relatives from Wisconsin would say, you betcha. Absolutely. We are still interested in beads. The difference is affinity chromatography is kind of like ion exchange chromatography. But rather than having a charged molecule, we've got a small molecule like glucose immobilized on that bead. Okay. Um, in the event that our protein gets stuck on that ligand, which is immobilized on our bead, it's going to stay there. It's going to stay there until you give it a reason to leave. So the this item right here, does the protein bind to the bead? That's the question. Yes? Okay, the protein is stuck. No? Move straight through. The bead, if it has a charge, that's kind of just the coincidence. That's because the ligand has a charge. Whether or not the protein sticks to the bead depends on the shape, attraction, and the complement of the protein. If the protein has an interest in binding to that ligand. So, for instance, if rather than hexokinase, let's say we had a mixture of hexokinase and HB or hemoglobin, and we had a bead right here that had glucose on it. Well, what's hexokinase going to do if it sees that bead? It's going to lock it down, set up shop there. What's hemoglobin going to do if it sees glucose? Doesn't care. Hemoglobin doesn't bind glucose with any sort of affinity of significance. How do you get those proteins eluted? Well, what you're going to do is you're going to add a solution that has that free ligand, that has that glucose free in solution. So then all of a sudden, this HK right here, which binds glucose and is currently bound to glucose, it's going to see this solution running past. And it's going to be like, hey, I love binding glucose. Oh, sweet. Oh, cool. There's glucose. Oh, man, it's kind of like that. If you've ever seen that meme where the guy is walking with his girlfriend and he looks back, it's that. The guy is our protein. I could make that and become a millionaire. Crap. Okay. So that's the principle of affinity chromatography. A ligand gets immobilized on a bead and a protein binds to it, and it stays there until you give that free ligand in solution, kind of flowing through. Or alternatively, you could also change the pH. If you change the pH, that's going to change kind of all of the different interactions within the tertiary or quaternary structure of a protein. Whenever I say tertiary or quaternary structure, it's just like, man, I feel like this semester has been like 14 weeks long already. I've appreciated it, and thank you guys very much for coming. But I can imagine that you guys are you're getting a lot of material. I hope that you're doing all right. You may have said the answer and I missed it, but is the type of chromatography that we do up to the discretion? That is a fantastic question. I did not say that. I'm happy that you brought that up though, because um, I think when people hear about affinity chromatography, they think to themselves, "Oh, that's the one to do. That's the good one." Um, it's very targeted, but it you know, basically anytime you're trying to purify a protein, chances are you're going to start out with homogenization and then salting out. That's kind of like a good way to clean up it's a very crude cleanup. After that, though, sometimes you might go straight to affinity chromatography. Sometimes you'll do ion exchange chromatography, then another round of ion exchange, and then affinity, then gel filtration. It all depends on what you're trying to do. If you, as a graduate student, I worked on a plant protein expressed in bacteria, so a recombinant plant protein, and I didn't have to worry about any sort of how clean my protein was. So I went from growing the cells, homogenization, salting out, and then straight on to affinity chromatography. If you're doing structural biology, though, you've got to get your protein like 99% clean. So that's a fantastic question. Thank you for asking that. Okay, our last separation technique is ah, one of your classmates. Dang, no. One of your classmates is going to become the millionaire who shared that meme with me. Okay. Um, so we've got hydrophobicity, and this is just, well, the there are regions of proteins that are hydrophobic. And this is just a little bit of a model where we have a hydrophobic molecule like one of these over here that's immobilized on a bead. And that's going to attract a hydrophobic region or portion of a hydrophobic portion of a protein. So that's kind of all there is to that. Okay, we've gone through a lot with respect to separation. We've talked about homogenization, salting out, and then 4+ different types of chromatography are 2 different ion exchanges, hydrophobicity, affinity chromatography, and gel filtration chromatography. So those are all just separations. Next up is how to analyze those samples that you're working with. Well, when we're talking about analysis, what we're really getting after is how effective were our separations. Um, did we clean up our sample? Did we get rid of 95% of the other stuff in there? Or did we get rid of only 5% of the stuff? Now, one of the ways that we analyze it is with SDS page. The molecule at the top here is known as sodium dodecyl sulfate. Okay. Dodecyl, I think is, let's see, 12 carbons. It's a 12 carbon chain with a sulfate and a sodium ion. And what that molecule does is it basically coats a protein and helps denature it to give it a uniform negative charge. Now, SDS page is a technique that really it allows us to separate molecules based on their charge. We're separating proteins based on charge, but we've given our proteins a total negative charge, a uniform negative charge. Why would we want to do that? The reason that we want to do that is because we take a protein, let's say it's 50 amino acids, and then another protein that's 700 amino acids. And when we expose it to SDS, those 50 amino acids effectively become 50 negative charges. Those 700 amino acids effectively become 700 negative charges. Those 2 different proteins, well, we've eliminated the variable of how different their charges can be. So that allows us to separate them. Yep, we need that SDS to help these migrate through the gel. Those negative charges, those thing, we're normalizing the negativity of these proteins. So then I'm going to say 50 and 700. So rather than, oh, what does this protein look in 3D shape? Instead, we can separate them based on those charges or based on kind of the magnitude of those charges. This one has 50 negatives. This one has 700 negatives. Now, what this shows us is a vertical SDS page. PAGE stands for polyacrylamide gel electrophoresis. This light blue gel right here that you have a little bit of a cross section of is made up of acrylamide. It's a polymer known as polyacrylamide. And what this ends up looking like, I remember one time I saw an image of like, it's basically like a spider web, a dense network of all these different layers, and proteins are going to migrate through there. You're going to load your protein sample up here, and then you're going to apply an electric current. At the top, it's negatively charged. At the bottom, it's positively charged. You've given your protein an overall negative charge. What are those molecules going to then be attracted to? Booyakasha! The positive charge at the bottom. So that 50 amino acid protein, oh, it's going to migrate through. It's going to be very attracted to that positive charge. Equally as attracted as that 700 amino acid molecule. The difference is, well, that 700 amino acid long molecule is 700 negative charges. It has to migrate through, weave its way through that network of basically spider webs. So at the bottom of the gel, your smallest proteins are going to be found. Because your smallest proteins are going to migrate fastest. I'm going to add this smallest equals fastest. At the top, your largest proteins are going to be left behind. This right here is a 200,000 Dalton protein. Or 200,000, that would be kilodaltons. So 200,000 kilodalton protein, a massive protein. It's going to migrate through, and in this first lane that I'm showing you guys here, well, these are what are known typically as MW or molecular weight standards. This is something that you have purchased. You know the size of these proteins, and you can look at basically how far do those proteins migrate from where they started, and how does that relate to their size? If you've done any DNA gel electrophoresis, the same principle is true. The size or the length of the DNA molecule is related to how far it migrates. So your smallest proteins are going to migrate the farthest and the fastest. Now, when you use those molecular weight standards that I've got set up here on the left, what that allows you to do is compare that to an unknown protein. So this is where you would be able to look at, okay, well, I've gone through 15 different chromatography steps, and I'm going to load my protein. Do I have a protein that's approximately 35,000? No, I guess that is Daltons. 35,000 Daltons. Well, and second, is my unknown protein the only protein in that gel? Well, let's see. Okay. First question, is there only one protein in that gel? In that unknown lane? That's lane number 2. Yeah, there is. Okay, that's encouraging. Next question is that protein 35,000 Daltons? And you can kind of eyeball that and be like, pretty close. Maybe it's 36.5. I don't know exactly. But maybe it's 37. It's kind of close. Does anyone think that there's a way that we could make a more precise calculation? Yep. 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 You're absolutely right. What we can do is plot the molecular weight of our molecular weight standards. So the size of the proteins in that lane number one. And then we can look at and compare the mobility. Basically, how far did those proteins move relative the final protein? Um, so that's one of the things that we can do. And from this, we can come up with a nice, yes, so just 1 second. So we can look at how far our proteins migrate from where they were loaded. Okay, our mixture was loaded right here. And so this protein, this first fat band there, it migrated, let's say, 2.2 centimeters. And then our last band, this goes back to, you do this with paper chromatography, your RF calculation. It's the relative migration. So this would be, okay, this moved 2.2 centimeters, and this last one, or my leading die, went like 14.6 centimeters. Is this something that I'm going to ask you questions about on the exam? Probably not. I wouldn't put money on this. Um, but what you can do is come up with a ratio of, well, how far did this first band move relative to the amount of time that I let it run? That's where we get our x-axis, our relative mobility. And then we say, okay, this 200,000 Dalton protein moved this far. We get a y equals mx plus b, a linear regression curve. And then we can plug in, well, how far did this protein move based on that? And then we would be able to make an approximation, a precise approximation that this protein is 36,500 Daltons. That's kind of the name of the game there. Okay, now there we go. There's yep. Okay. The last thing that I want to mention is, what about disulfide bonds? So the SDS is going to help us denature our protein, but we got those disulfide bonds. We have to break those disulfide bonds. Cysteine bonds. Well, if you remember, we talked about this with Christian Anfinsen. We use beta-mercaptoethanol to break those disulfide bonds. That's significant because if you have a non-reduced protein, it might run all the way up here. A multimeric protein kind of running together because you got a disulfide bond between them. Lo and behold, this right here is not one protein, but instead it's two proteins that are approximately this and this combined. With the addition of the beta-mercaptoethanol, we broke those two things apart. Um, and this is my stopping point. Yeah, so this is where I'm going to stop. What I will reiterate is, if you want to meet and go over something tomorrow, Friday, Monday, Tuesday, Wednesday, I can do that. This is one of the luxuries of having a 6:30 p.m. class. I'm going to go ahead and just a second.